Let's share the screen and we'll get right into it. Okay. So, scoping out the chapter a little bit, it's going to talk about how our bodies are basically these microbial menageries of complex communities, as we kind of talked about a little bit last week. Back and forth and a collaboration between us and these microbes. And, while most of them actually don't do much to us unless they're opportunistic and can find a portal of entry, a lot of them are beneficial and help us with our digestion and nutrient acquisition, as well as shoring up our immune system and making us primed for other pathogens that will infect. It can also help us when we are colonized with good bacteria or other good microbes. There'll be this antagonism between those microbes and the bad germs that want to seek residence in those areas. So while there are pathogens, there's also a lot of microbes that are helpful to us. And we'll talk about that a little bit. We'll talk about the infection portals and pathways. So where these microbes gain access into our bodies and what they do once they're inside, okay? And which areas in our body is it common to have microbes, and which areas is it supposed to be sterile? And when it's not sterile, we have then infection. We'll talk about some of the adaptations that these invading microbes have evolved in order to gain access into these areas where our body wants to keep them out, whether it's the structures that they have. And we'll go into more of the anatomy of particularly bacterial cells later on. So we'll kind of gloss over these structures, but they have hooks and like fibers that help cling on like Velcro to the cells. We'll talk a little bit about that. And then once the pathogen does take up residence and starts replicating, what are the results of the toxins and the enzymes that it's releasing to help it gain access in terms of its manifestation in us as disease? And subsequently, if it's chronic enough, damage to our tissue. And then finally, we'll get into the way that microbes travel between hosts, right? And that's something that is percolating in everyone's mind right now as we talk about COVID-19 and whether or not to wear masks, whether or not to be socially distant, right? Because it's a respiratory disease. Well, it's a disease of more tissues than just the respiratory, but that is the way that it enters into the body, okay? Okay, so clearly, as we've learned, we are not alone, right? We are covered in microbes because microbes are ubiquitous, as we've said. And for most instances, they are protective to us and they stabilize the physiology of our bodies. However, when they're not doing that, when they aren't helping our immune system become more robust to the bad guys, they are the ones that are causing damage. Okay, and that's when they go into places that should be normally sterile, like our lungs. So to get into a few definition terms, when we talk about microbes that engage in mutual or commensal associations, those are our normal resident microbiota, right? So commensal, if you guys have had other biology classes, you know that symbiosis can be parasitic parasitic, where one host is damaged and one host benefits. It can be mutualistic, whereas both of the species benefit somehow. It can be amensal, which is really kind of a cruel form of symbiosis where one organism is hurt or harmed and the other species is not benefited or harmed at all. So it doesn't get anything out of that species being harmed. And then there's commensal, where one benefits and the other isn't harmed. Okay, so mutual and commensal symbiosis is nice, right? Because either both backs are getting scratched, or one's getting scratched and the other one's, is fine. And that's what our normal resident microbiota are. They're mutual or commensal, and it can be thought of as indigenous, meaning it's native to our flora. You might hear it as microflora or normal flora, commensal. These are all interchangeable definitions to describe microbes that are beneficial to us, or at least not detrimental. A pathogen, on the other hand, is a microbe that causes infection. Okay, so patho is basically disease or abnormality, and gen is to produce. So a pathogen produces disease. And when it does, that is classified as an infection, okay? It's when the pathogenic microbes break beyond your defenses, enter your tissue, and then start reproducing. And infectious disease is then going to be an infection that causes damage to your tissues and organs, okay? So we can have lots of infections that our body then clears and we feel uncomfortable for a few days or a week, but really doesn't damage our tissue. But when you have an infectious disease that is a chronic infection, then, or maybe not chronic, but just a really gnarly infection, like measles, that's going to then disrupt your tissue, and it's going to be a disease as a result. Okay, so where do microbes take up residence in our bodies? Well, there are areas on our skin, obviously on the external parts of our body, that's going to be very, very likely to have microbes. But our internal organs, in our blood, in our other fluids, should be microbe free. In general, we don't have any microbes there, and if we do, then we have an infection. When we talk about this, we can have microbes that are transient, meaning they're in our bodies for a period of time, and then residents, meaning they take up full-term lodge and lease in our body. Okay, they're established. The skin is going to have things that are both transient and resident. Transients are going to be the things that when you shower and wash off, they go, and then they come back, and then you wash them off again. But there's not a real niche there for them to cling to and stay there in the long term. So transients are going to be influenced by your overall hygiene. Whereas residents, they are going to be less influenced by washing. So they are more impervious to the hygiene process. And this is going to be a lot of bacteria and yeast. And then there are little tiny microscopic mites that are in all of our faces. I don't know if you know that or not, but we have these little tiny microbes in the pores of our face that are just always there hanging out. And luckily we can't see them, because I think it'd freak most of us out if we knew just what was living in our skin. These are going to be stable microbiota. It might fluctuate again depending on your hygiene, what you eat, hormones, and if you're taking antibiotics or other drugs that might be harmful to them. But overall, these are going to be beneficial. And I mentioned a little bit earlier the microbial antagonism. The nice thing about that is when you have microbes that are good for you, or not harmful at least, take up residence on or in your body, it makes it harder for the harmful microbes to take up residence there. They have to be able to kick those good ones out. Okay. And so they provide this thing called microbial antagonism, where it doesn't even need to be your own immune system fighting it. It's surely the microbes that are good for you kicking these bad guys out. Okay. And then endogenous infections. That is a type of infection where your normal flora goes to a place that is sterile. Okay. So you might have heard of leaky gut. And, there's not a whole lot of great science, I think, on the mechanism of leaky gut and what influences it and to what degree it exists. But the idea is that you have your gut microbes and they are living inside your digestive tract. But if they start penetrating through that mucosal barrier into the more internal areas of your stomach, then they're going to get introduced to places that is not used to having microbes, and that can cause inflammation. That can cause also infection. So that is an endogenous infection. It's not when a microbe outside finds a portal of entry. It's when the good guys go where they shouldn't be. Here's just a list of the organs and tissues that should be sterile. All right. So your heart and your blood, right? When you have microbes in your blood, now you are septic. Okay. So we really want to keep our blood germ free, because that goes everywhere. So once it goes to your blood, now it's no longer localized, and it can go and take up residence in other tissues and organs. Your lungs, you don't want bacteria or viruses in your lungs. When you do, then you get things like pneumonia and stuff. So it should generally be free of microbes, good and bad. So when I'm saying sterile here, that means nothing there. Not good, not bad. Your bones, your sinuses, right? When you get a sinus infection, it's because you have microbes there. Middle ear, inner ear, you get an ear infection, right? So all of these things should generally be sterile. And when they're not, that leads to infection.
So Carl, quick question.
Yeah, sure.
Since skin wasn't on that list, would they play a role in acne?
Oh yeah, definitely. So skin is not on the list that should be sterile, because there's no way you could really sterilize your skin. But and we'll talk about that later when we go into specific bacteria and what they do. But in terms of acne, there are microbes that can cause like things like folliculitis, so infections of the follicles. So not necessarily like, so with acne, you have your pores getting clogged from sebum and wax, and then that way it can't flush out these microbes, and then that's how those things get infected. So yeah, absolutely.
Interesting.
Yeah. And we talked about hormones being one of those things that can regulate the your microflora, right? And so definitely, if you're producing hormones that then cause your pores to clog up, then they can't flush out that bacteria as well. It gets infected. So this is looking at floral species that are in areas that aren't supposed to be sterile, that couldn't be sterile unless you lived in a sterile bubble. Your skin, Pseudomonas and Streptococcus and Staphylococcus. And Pseudomonas is an opportunistic pathogen. It's not necessarily bad, but if it is able to get inside your body, then it can be. But these are going to be mostly in the upper layer of your dead skin cells, some of the follicles, things like that. Here's the Demodex mite that I was talking about, this picture here. This is a little guy who lives in your like hair follicles on your face. And most of us have them. I think not every single human being has these critters, but most of us have them in our face right now. The GI tract, clearly there's going to be lots of different bacteria, and the Bacteroides was one, and the Actinomyces that we had talked about in that paper. That is a really common bacterial species in the gut. Here's Veillonella, if you remember that, right? That was the performance enhancing microbe. And then we have lots of fungi. The Candida albicans fungi is opportunistic also. It's all over us, and it doesn't really harm us unless we are, we're immature, we're young, like infants, and our immune system is not mature, or we have some kind of immunocompromisation that can't fight off the Candida. Your large intestine and rectum are going to have a bunch of different bacterial species. And these tables, I don't want you guys to worry about memorizing the different bacterial species for each region. Okay, at this point, that's not part of what I want you to concern yourself with. Just be aware which tissues are established with microbes and which ones aren't. And we'll get into the different species and things like that later on. So don't overwhelm yourself memorizing these tables. This is just kind of an informative overview of the different parts of our bodies and what we can expect to find there. Urinary tract, eye, ear, most of these things are going to be bacteria. There aren't really viruses that are here that are normal, beneficial residents of our body. If you have a virus that is a long-term, well-established microbe, that's not a good thing, right? So all of these things are going to be typically bacteria, some fungi as well, right? So when babies are developing in the uterus, the fetus is going to be sterile. And unless the mother has some kind of infection or something like that that is able to cross into the fetus. But typically it's sterile. And then once the water breaks, now it's game on, and that baby needs to get out as soon as possible, because the longer it's in there, the longer it's going to be exposed to microbes, because once the water breaks, it's no longer protected. So regardless of whether or not there is a C-section or the baby comes out of the normal birthing canal, it's going to be colonized with different microbes on its way out. And there's a lot of debate on whether or not, C-section babies should be exposed to the natural flora of the mother's vagina afterward, and because it's very different microbes inside there than coming out of the stomach. And how does that relate to things later on in terms of their overall health and their microflora? Does it change based on the way that the babies are born? And I haven't really seen a whole lot of rigorous data saying that there's really much of a difference. That essentially the babies come out, and as they're being held and exposed to their parents and family and friends, they're going to get colonized, and they'll catch up regardless of the way that they are born. Now, it has been shown that whether or not babies have milk or formula will drive certain bacterial species in their gut. Because remember, milk has lactose in it, and lactose is a sugar that certain bacterial species feed on. And so depending on the diet of the baby, that will drive an abundance or a lack of abundance for certain bacterial species based on the nutrients that that baby is encountering, which is interesting, I think. Any questions so far on this? All right. So your microbial residents in your GI tract are going to be based on the conditions of your GI tract. So the pH, the overall anatomy, if you have blockages, right? Or if you have areas where they can't get to, then that's going to be an issue for them. These areas include your oral cavity, your intestine, and your rectum. And there's going to be lots of microbes in those areas because, for one, your mouth is open a lot, right? So it's just a huge portal of entry. And the large intestine is where all of your food gets processed and broken down. And so there's all these nutrients that are rich and ready for those microbes to then process and feed on. Like I said, the mouth is going to be the most diverse, okay? Because it's just, that's where everything goes, right? And if you're a kid, you know, like your hands go in your mouth all the time and your hands are touching everything. And so between that and then all the food that you eat, your mouth is just a very, very robust source of bacterial growth, which shouldn't come as a surprise, right? You have all different types of niches. So when we talk about a niche, we're talking about like a place where it lives, right? Whether it's the epithelium inside your cheeks or your tongue, which is going to be a very different source of niche than the epithelial cells. The floor of your mouth, your teeth, right? There's all these different structures that can be favorable for a particular flavor of bacteria. And what we see usually are these streptococcus strains, sanguis, salivarius, and mitis. Of course, when babies are cutting teeth, now that tooth is going to erupt through the gum line and open it up, right? So now you don't have that protective barrier, and you can get anaerobic bacterial species now coming inside into the gum line. And anaerobic meaning they don't need oxygen. And then that's what's going to start causing things like periodontal disease, particularly. And cavities to some extent, but cavities are also on the outside on your teeth as well. Does anybody know how bacteria causes cavities? Or did you know that bacteria is what's caused the cavities and not just sugar?
I thought bacteria caused gingivitis, and I didn't know what cavities were caused by.
Okay, yeah. It does cause gingivitis, and it causes cavities because what's happening is that, and it is as a result of having too much sugar and drinking a lot of juice, because that sugar feeds the bacteria in your mouth. And as a result, they eat the sugar and they break it down into acids. Remember we talked about propionate and acetate and butyrate in that paper, that Journal Club paper last week. So the bacteria excrete acid as a byproduct of breaking down all this lovely sugar that you're giving them. And the acid is what breaks down your teeth and gives you these cavities. And a cavity is essentially just like a hole in your teeth, in your tooth, where a bunch of bacteria was just, deposited, and usually like in the cracks where you're not flossing, and it secretes the acid, and that just wears out your teeth. So that's how you get cavities. Blame it on the bacteria. And there's a lot of bacteria. So in 1 milliliter of saliva, which is, not much, 1 mil, you can have, was it like 5 billion bacterial cells in just one little mil of saliva. So they're everywhere. Your large intestine is also going to have tons of bacteria. So even more, on the order of 10 to the 11th microbes per gram, which a gram is about basically a mil for solids. And about a gram of feces, you're going to have even more microbes than a mil of saliva. And these are the common flavors here, the Bacteroides, Bifidobacterium, Fusobacterium, and Clostridium. Now, these bacteria that are good for us provide, again, all kinds of different benefits to us by generating byproducts in terms of vitamins. So a lot of the B vitamins, not all the B vitamins, but a lot of them, vitamin K, these acids. So remember, here it is, acetic acid, butyric acid, and propionic acid. The propionic acid, or the propionate that we had talked about, is what they think is allowing these athletes to, and the mice particularly, to have longer endurance. So it's beneficial from that perspective. Also, they have enzymes that can break down the sugars in our body, the disaccharides, so 2 to monosaccharides. They can promote steroid metabolism. They can break down fibers and other organic matter that our bodies cannot. So, lots and lots of benefits that if we did not have any microbes in our gut, we'd be in pretty sorry shape. Respiratory tract. It's fine to have microbes in your nose, in your larynx, but once it gets down into the lungs, then that becomes bad. Okay, we don't want things in the lungs or the bronchi. All right, these branches here. But anything above that is going to be well colonized with things. And again, it's because of that openness, that easy portal of entry for microbes. And this can be anywhere, anything like, so in your nose, you're going to have Staph aureus, which can be bad. And in the mucous membranes of your nasopharynx, you can have Neisseria, which can also be bad. And then in your tonsils and lower pharynx, you can have a bunch of different types of streptococci and Haemophilus. So some of these are fine, but can be bad if they go beyond your respiratory tract, right? If Staph aureus then goes, beyond that inside your body, then it can start being very, very bad. And so,
what prevents them from actually getting down into your lungs? Because your airway's fairly open with your epiglottis all the time.
Yeah. So typically, it's mucus and these little microvilli that help keep it out. So it brings the bacteria and other microbes. It sticks in that mucus, and then it kind of like conveyor belts it back up into the larynx. And that's why when you do have a lower respiratory infection, you get a cough and you get a lot of mucus buildup, because it's the body's way of trying to trap all those microbes and get them out of the lungs. Any other questions? Okay. So yeah, Staph aureus, that's bad. If you guys know about MRSA, right? It's methicillin-resistant Staph aureus. So bacteria can be bad or good depending on where it is in or on your body. This is kind of just reiterating the same stuff. Normal flora is good. And antibiotics, changes in your diet, overall disease, alter that flora. When you take a bunch of antibiotics, sometimes you have to, right? But if you're taking it and if you're not taking it to the full duration, which we'll talk about later on, why it's important, if it says to take it for 2 weeks, that you don't stop taking it when you start feeling better. That's a different topic. But when you take antibiotics, that's destroying the bad guys, but it's also destroying the good guys too. So it's going to change your flora. And remember what we said about the antagonism. If you are wiping out the good microbes, you're now making that kind of a carte blanche area for bad germs to come in and colonize. So while antibiotics are important and need to be used, they should be used with discretion. Probiotics, that is introducing known or unknown microbes back into the body. And we've kind of talked about that recently, about what are the true effects of probiotics. Is it really efficacious in a normal person who's not experiencing certain illnesses and heavy antibiotic treatment? But that's the concept anyway. And then prebiotics, those of course are going to be the things that you eat that help the good bacteria. So a prebiotic of sugar, while that's not good, it's a prebiotic for your mouth bacteria and your gut bacteria too, for that matter. Okay, so what do you guys think? Normal resident microbes are absent or should be absent from what? The pharynx, the lungs, the intestine, hair follicles, or mouth?
Lungs.
Lungs.
Yeah, absolutely. Lungs. All these other places, there's going to be lots of microbes. All right. The major factors that lead to the development of infection are right here. Portal of entry, getting in, attaching firmly. So once they get in, they need to attach to a cell to infect it, right? And with SARS-CoV-2, we know that that is the ACE2 receptor that it binds to. Once it attaches, it has to survive the host defenses. So it has to survive cytokines and chemokines that are excreted to poison these things. It's got to survive the phagocytes that eat them, like the Pac-Man cells that will try to eat them. It has to survive the antibodies that were previously developed to target them. And once it's attached and survived and entered, then that's when it's going to cause damage and disease as a result of it doing its thing, really, right? The microbes just want to reproduce. That's their main goal. Their goal isn't to make us sick per se. That's a process that happens by proxy. And then, of course, once they've set up shop and churned out a bunch of babies, they need to exit the host so that those babies can go do the same thing to some other host, right? So those are the major factors. These are the major factors involved in infection. And if you take out any one of these steps, you inhibit further infection, okay? And so when we later on go into the drugs that are developed to prevent infection, some of them will be at different stages of this development pathway, right? Some will prevent them from entering. Some will prevent them from attaching, right? It would be nice if we could have an antibody that would just block the ACE2 receptor on our cells so that the SARS-CoV-2 couldn't bind to it. Now, the problem is that the ACE2 receptor regulates your circulatory system and vasodilation and stuff like that. And so you can't do that because that would make you severely hypotensive. But there are other antibodies that are used to block certain receptors from viral attachment. And so on and so forth. So we'll get into each of these in a little bit more depth finding a portal of entry is going to be your skin, the GI tract, right? You eat something, respiratory tract, urogenital tract, particularly with STDs, and then endogenous biota. Remember, the endogenous microbes go places where they shouldn't be. They use these different structures to attach. And again, we haven't gone over these structures, so right now I won't spend a whole lot of time, but the fimbriae, capsules, surface proteins, viral spikes. So again, with SARS-CoV-2, it's using its spike on the outside that gives it that corona appearance that looks like the sun to attach to the ACE2 receptor on your cells. And then certain microbes will also have just like hook structures. So they hook on like Velcro. Surviving host defenses, that's going to be in the form of avoiding phagocytosis, like I mentioned, the engulfment of the pathogen by an immune cell. Or there are other strategies where it can get engulfed by the phagocyte, but avoid subsequent breakdown once it's inside that phagocyte. So it's armored to protect itself from the enzymes in that phagocyte that would break it down normally. And then, of course, evading actions of the immune system, somehow signaling that they are not bad guys and that they shouldn't be targeted for destruction.
Aren't there some aren't there some pathogens that actually like being eaten up by cells, like HIV? Don't they actually reproduce inside of a cell?
Yes. Yes, all viruses will produce inside a cell because they need the machinery of that cell to reproduce. They don't have their own ribosomes, and so they can't translate their genome into proteins. So yes, in fact, but the phagocyte is a, and we'll talk about that in the next lecture or two. The phagocyte is a particular immune cell that is responsible for eating and destroying pathogens. Now, HIV, like you mentioned, that infects our immune cells. And so not phagocytes in particular, but that's why it's so deadly, and that's why you end up dying not from HIV but from AIDS, because the HIV is destroying your immune cells. Those immune cells then can't go and fight off other things, other opportunistic pathogens.
I was going to say, you don't actually die from AIDS, you die from a secondary infection, don't you?
Yeah, exactly. So you die because your immune cells have all been obliterated by the HIV, and so then something like cryptosporidia, or another co-infection with influenza or anything really can come in, and whereas a person without AIDS and a robust immune system would clear that infection, or it wouldn't even be able to infect that person. When you're compromised, you can't fight it off like you would normally. And so they end up succumbing to that. And then the causing damage and disease, they're going to be releasing toxins and enzymes. Lysis is breaking down your cells. And so that's directly linked to damage. There's also indirect damage where the host response is inappropriate and excessive, meaning the cytokine storm that is released causes damage to the tissues of your body as well, not just the bacterial cells, or inflammation. Or once you have your immune system, think of it as an army, right? It's mobilized and it's primed and it's destroying the bad guys. For some reason, it doesn't get the signal to then back off, right? And then it just keeps going and it starts destroying its own cells unchecked. And you have autoimmune disorders as a result. So So the damage can be from the bacteria doing it or from our own bodies not knowing how to regulate and balance the attack. And then exiting the host. Again, with COVID-19, right? We know that it's the respiratory tract that is releasing new viral particles that have been made there to then enter into someone else's respiratory tract. Salivary glands, skin cells, fecal matter, right? If you have poor sewage treatment and sanitation, then there's all kinds of disease that can manifest by having all of this poop around. The urogenital tract, blood, right? And blood should be sterile for all intents and purposes, but when it's not, we get blood-borne pathogens, which are a huge issue, right? Blood-borne pathogens are bad mostly because they are microbes that have adapted to be in a place where they shouldn't. Okay, any questions on that now? Any other questions? So we've talked about the good guys and how they provide us benefit and how we provide them a home. Now we're kind of phasing into the bad guys and how they take up residence in us. So again, dealing with what's called phase one or the portals of entry, there are going to be exogenous agents that come from outside the body and endogenous agents. So exo, outside, endo, inside. Already, and so that's, again, your normal flora. So an endogenous, if I said, what is the difference between an exogenous agent and an endogenous agent? You would say, well, an exogenous agent is going to enter through a portal of entry externally, right? Where an endogenous agent is already inside, but it has to find a portal of entry internally, going somewhere where it's not supposed to be. And all of these microbes have what's called an infectious dose, or an ID. The minimum number of microbes required for an infection to proceed, okay? It's not that you just get one bacterial cell or one fungal spore or one viral particle and now you have infection. And with each microbe, that number can be low or high depending on the microbe itself. And typically, if it's highly virulent, so causing a lot of damage, much more likely to be detrimental to you physiologically, it's going to often have a small infectious dose. Okay. If you don't reach that ID, you won't get an infection. Plain and simple. And this is a table of the infectious dose. And this is not something you have to memorize, but I'm putting this up here to give you an idea. With measles, one viral particle, and you can get infected. And that's why measles is so contagious. And that's why we see these outbreaks at Disneyland and stuff, and why it's terrifying, because you only need one viral particle to enter your respiratory system, and now you have it. If you haven't been vaccinated, or you're an infant and haven't been vaccinated yet, you could be in major trouble. Whereas with smallpox, you need anywhere from 10 to 100 cells going into your respiratory system in order to get an infection. Cholera, at the very bottom here, you have to ingest 100 million cells, which seems like a lot. I mean, it's not a whole lot, though. When we talk about the fact that there are billions of cells in 1 ml of saliva, right? And in 1 gram of feces, there's billions. So, but still, you have to have much more bacterial cells for cholera than you do viral particles for measles. And each disease is going to come from a microbe that has that requires a certain number to take residence. Okay. Phase 2, so they've entered, now they have to adhere. They have to stick to the host. Otherwise, they'll just get passed right through. And they use these different structures to do so. And again, we'll be talking about all of these structures in further detail. I mixed up the order of things for this class because I wanted to get to COVID sooner than later. Typically, I talk about these chapters later on, and so we've already talked about flagella and the glycocalyx. But since I moved it up, I'm not going to go into these specifically, but know that these are some of the examples of things that they use to stick to the cell. This is, again, not something you have to memorize, something that you can review if you want, but it shows the different types of microbes and what disease they cause and how they adhere. So HIV causes AIDS. It uses viral spikes to adhere to blood, to the immune cells. E. coli causes diarrhea and other things, and uses fimbriae to adhere to intestinal cells. Okay. So they all have their different things that they've adapted to use, but it's the same goal, sticking to the cell and then entering in. Phase 3, now, once they've entered, they are going to become established, okay? And let's take a 10-minute break here because it's 10:50 now, and we will finish up the lecture at 11, starting at 11, and then we'll get into the journal club, okay? The Henrietta Lacks reading. So any questions before we take our break on that material that we've covered so far? Pretty straightforward.
I just had a question on the footholds of certain bacteria. Why does it take so many cells sometimes for something? Is it because how quickly our body can take care of it, or what?
Yeah. So the 100 million cholera cells is because that's the amount that's needed to overwhelm our immune system and not be able to clear it beyond that.
So would that number be lowered if our immune system has been compromised?
Certainly. Yes, great question. That applies to someone that is immunocompetent. But certainly if you, and that is exactly why people that are immunocompromised, because they have, for instance, HIV, let's say, that is why they succumb to these pathogens that don't normally kill people, because the infectious dose now is way lower. And so they can take hold. Any other questions? Okay. Well, I'm going to pause the recording. Remind me to unpause if you don't hear that little voice saying this is being recorded. Let's take a 10-minute break, leg stretch, and then we'll get back into it and wrap it up and then get into Journal Club. Okay? So there we go. Recording again now. I don't want to forget that. Let's share the screen. Where did we leave off? We left off here. Right? Right. Okay. So now we're in phase 3. We've talked about portals of entry. We've talked about adhering. And now we're going to talk about establishing and infecting. Okay, so the bacterial cells will then secrete these enzymes that help break down the barrier. So if it's your epithelial layer, for instance, it will break down the membrane or the mucosal layer. Oftentimes it's like your mucosal layer. And that will allow it to then penetrate beyond that protective barrier. They can secrete toxins. The bacteria will secrete toxins that will then target the cells and will kill them. so that they can gain access further into the tissues, or so that they can be released from the cell if they have entered inside of it. And then that starts, once your cells start becoming damaged and die, they slough off. And so, you can think about Helicobacter pylori causing ulcers, right? It's because it's damaging the epithelial cells and causing a sore there.
Is this what necrotizing fasciitis does?
Yeah, exactly. It damages, it releases toxins that kill off the cells, your cells. And so you get necrotic tissue as a result. We mentioned blocking the phagocytic response, right? So either they don't get targeted by these Pac-Man cells at all, or they get engulfed, but then they don't get broken down inside the cell. They've evaded. So once it gets inside the cell, then it will release the host cell will release certain enzymes to break down that microbe. And it has evolved ways to protect itself from that. Different microbes have different ways of being engulfed, and this is a invasion factor from Salmonella, so it's pretty cool, actually. The Salmonella binds to the cell by fimbriae, and then it releases these proteins. It's kind of like a filament, and the protein chain goes down into the cell here and targets these actin filaments, these hair-like things. Actin filaments, they polymerize and they lengthen, and that's what helps cells move, the motility of cells, right? And it's like, so this protein chain tickles those actin fibers, and they polymerize and lengthen, and now the edges of that cell engulf the Salmonella cell, which then divides, multiplies, and releases itself. And different microbes, again, they all have their different ways of getting entry, getting attached, and setting up shop. In terms of toxins, there's all different types of toxins and different types of reactions to those toxins. When we talk about toxinosis or toxinoses, that is just going to be the effects of those toxins on the host. If it's a toxemia, that means it has gone to the blood, okay? So you have a septic infection, okay? And now the toxin that the bacteria is releasing is not just released from the cell, but it enters into your blood, and then from there, your blood takes it throughout the body, and now you have a systemic toxic reaction. Whereas intoxication is caused by the ingestion of the toxin, okay? So it's not endogenous, it's that you are consuming the toxin from these cells that were in some food, let's say, or like Botox, right? If you have Botox, you are injecting, not ingesting, but you are injecting that toxin into your skin. You have exo and endotoxins. Again, an endotoxin is not secreted into the cell to then break the barrier up, but it's released after the cell has been damaged. All right, so the toxin comes from within and then is released upon damage of the cell. Whereas an exotoxin, remember exo is out, endo is in, that is secreted by the bacterial cell into the tissue to infect, to allow it to take residency and infect that area, okay? And there are different types. Hemolysins will then disrupt red blood cells. There are AB toxins and the A is the active, B is binding. And we'll get into these different types of methods later on. So don't worry specifically about these. Just know the difference between an endotoxin and an exotoxin for now.
Could an endotoxin be either for the cell it's trying to invade or the bacterium itself when it's released, like when it dies?
Say that again.
So, like an endotoxin, could it be secreted after the bacterium itself dies, or is it just something it injects into the cell it's trying to invade?
Yeah, that's a good question. Typically, it's released once the bacterial cell is inside. But for instance, when we talk about this endotoxin being part of the outer membrane of the gram-negative cell wall, if you have dead bacterial cells, which you will have, right, especially the parent cells, then yes, it can be byproducts of the dead bacterial cells as well. Does that answer your question?
Yeah.
Again, another table. Don't worry about memorizing the table, but if you want to look over this to kind of ferret out the major differences between exo and endo and what they do, then this is a nice comprehensive table. It tells you that, for instance, the toxicity of an exotoxin is higher in smaller amounts, whereas with the endotoxins, you need a higher abundance of it for it to be as toxic. And then, different things that they're made of. So small proteins, and exotoxin is released by the bacteria as a protein, whereas an endotoxin is going to be a component, the lipopolysaccharide, or the LPS, of the cell wall. And an LPS is a lipopolysaccharide, is something specific to bacterial cells, and we'll talk about later, is a major trigger for the immune system. If it detects LPS, then it will start mounting a response to it. And so sometimes it's nice that these endotoxins are flags that are waving to alert the body to an upcoming infection. So I won't go through all of these things because I don't feel like you need to memorize it, but if you want to review it, especially, if you're going into nursing and stuff, this might be important. Factors that weaken the host defense and increase susceptibility to infection are, of course, things that are going to compromise your overall immune response or your defense barriers, right? So if you're old or young, you're going to have a less robust immune system. If you're young, your immune system is less robust because it hasn't developed fully. If you're old, your immune system isn't as robust because it's not churning out the B cells and the T cells as much as it used to, and the antibodies associated with those T cells that they use and the B cells. And all of that is going to lead to a weaker army, right? Genetic defects in your immune system in general, or acquired defects in immunity, like with AIDS. Surgery, organ transplants. Why is that an issue? Anybody know?
With a transplant, you have to have your immune system lowered just so that you don't reject it.
Exactly. So that you're going to be on immunosuppressants for a transplant. What about surgery? Surgery, you're cutting open your skin, which is a great barrier against disease, right? Against germs. So cancer, that's another one, right? If you're a cancer patient, you're getting radiation or chemotherapy, and that doesn't just target the cancer cells, right? So it also impacts your immune cells. It kills off a lot of things. And so you're going to have a weaker army. Your stress, the amount of stress physically and mentally that one person is dealing with can often affect their immune system. Right? And then, of course, other infections. If you have co-infections, you're going to be much more susceptible to a particular one than if you just had that infection by itself. Okay. Now, true pathogens versus opportunistic pathogens. True pathogens are going to be able to cause disease in healthy persons, regardless of their immune system status. Okay, so think of flu and malaria, all kinds of things. These are just a few examples, right? But those are not going to discriminate a healthy person versus a sick person or an immunocompromised person. Whereas opportunistic pathogens are only going to cause disease when the host defense is compromised. Okay, so whether that be from another infection like HIV, or from chemotherapy, or from immunosuppressants, things like that, or from surgeries like Pseudomonas. We said Pseudomonas can live on the outside of your skin, but if you cut yourself open, then it can gain access inside. Candida, that's a fungus that is all over us. It's all over everywhere, but it doesn't really usually affect people unless they have an immune compromisation of some sort. And then it becomes opportunistic. It's not a pathogen. It doesn't need, opportunistic pathogens don't need to destroy or cause damage in the host to survive. But if they're given the opportunity, they will. Virulence factors, those are things that contribute to the ability of a microbe to cause a disease. Okay, there's all different types of virulence factors, but it could be different structures that they have, or it could be different enzymes that they produce, or toxins, and that's going to lead to the overall severity of that pathogen. Are they destroying your cells when they exit, or do they just bud out of the cell without destroying it, right? There's a balance. Not all pathogens destroy the host to the point of being lethal. In fact, it's counterintuitive to kill the host because the host is what's needed to keep the microbe alive. So oftentimes it'll cause enough disease and discomfort to do what it needs to do, but not enough to kill it, because if it kills off all the hosts, then it dies with it. The process of infection is broken down into 4 different clinical stages. The first is the incubation period. Okay, that is how long it takes for this microbe to enter the host and start causing the first symptoms. Okay. And that's, when we're talking about, again, with COVID-19, the question is, well, you could have the virus and have no symptoms because there's an incubation period. And you can go to the hangar or, go to Aleworks and have COVID, but not know it and be infectious, right? It's the time that it takes to set up and then cause symptoms. Then you have the prodromal stage. This is like the vague feelings of discomfort. So, oh man, I don't know, today I got a sore throat. I don't know what's going on with me, that kind of a thing. And then once you get past that, you get into the period of invasion. And that's when you really start feeling like caca. You feel terrible, right? You see these signs and symptoms start to progress, and maybe you have a fever, maybe you have chills, body aches, that kind of a thing. You know that you're sick. The prodromal stage is, maybe I'm sick, I don't know, maybe I'm not. Period of invasion, you are sick. And then there's convalescent period. As it starts to wane and decline, your body ramps up a sufficient response to keep this pathogen in check, and you start getting better. Those are the 4 stages. Infection can be all different types. You can have a localized infection where the microbe is confined to one specific area, like influenza, right? Influenza infects your respiratory system. Or you could have a systemic infection where it spreads in the bloodstream, okay? So that would be like a septic situation, right? Where the pathogen is now traveling to other parts of the body via the circulatory system. And now you have systemic infection. These are bad infections. These are much harder to clear than a localized infection. And then you have focal infection, where an infectious agent goes from local to other tissues. And this is what we're seeing with SARS-CoV-2. Your ACE2 receptor is not just in your lung tissue, right? It's in your circulatory system. I believe it's in your liver, right? It's in these different spots. And so now you get focal infection. It's localized in your lungs and then it starts spreading to other places. It's not systemic because it's not really in your blood, but it's moving to other places that have that ACE2 receptor and binding and infecting those tissues. And then mixed infection is just having more than one infection at the same site. So it could be polymicrobial is another way of explaining it. Co-infection with more than one different type of microbe at the same area.
I'm confused how an infection or an agent breaks loose and is carried to other tissues without being in the blood.
Yeah, so it can travel in the lymphatics. So your lymph node, you have you have your lymphatic system. So it can travel that way. It can travel through your digestive tract and then enter through that way. Or it can be in the blood, but not to the point where it's causing havoc systemically, right? So it's in the blood and it transfers from, say, your lungs to your heart via your circulatory system, but it is not profoundly located in your blood to the extent that it's infecting, whole body tissue. Does that make sense?
Yeah, makes sense.
Yeah, thank you. That's a good point of distinction. That you can have focal infections carried to other locations via the blood and not be systemic per se. And if it's not the blood, there are other ways. Primary infections, secondary. I'm going to kind of move through this fast because this is pretty straightforward stuff. Primary is the initial infection. Secondary infection is now your immune system is being taxed because it's trying to fight off this initial infection. And as a result, other microbes join in on the party, and now you have a secondary infection by something different. Acute infection, it comes on rapidly but short-lived. Your body clears it and then you're better. Chronic infections last over a long period of time. They have maybe a latency period where you don't even know that you have this infection. Or that it's not really infecting, it's just inside and quiescent. And those are going to be chronic. Some are lifelong, some are not lifelong, but can be years or decades. Now, this is something I really want to focus on because we hear about signs and symptoms and categorically they're different, okay? A sign of disease is something that is objective, meaning, let's say you are a nurse and a patient comes in, you can look at things with that patient and have an objective determination of whether or not they might have a disease. Whereas a symptom is subjective. It's what the patient tells you, right? Oh, my throat is killing me, right? That is subjective. You always see those pain charts, right? 1 to 10. 1 means mild discomfort. 10 means you're like being mauled by a bear. One person's 9 is very different from another person's 9. It's subjective, okay? And so let's not mix those up. Signs are what the observer notes. Symptoms are what the patient is experiencing. And both of them have their informative benefits, but symptoms are harder to diagnose with a disease because, it's patient to patient. So if we look at signs, right? Fever, that's something that you can measure. You can measure someone's temperature, and if they have a fever, that's not subjective. Septicemia, you can take a blood sample and culture it and see what's there. And there are either microbes there or not, right? Abnormal chest sounds, skin eruptions, swollen lymph nodes. These are things you can observe, but you can't observe if someone says they have chills. Okay, that's what they tell you. You have to take their word for it that they're experiencing chills. Their discomfort level, right? Nausea, chest tightness. You can look at, you can observe abnormal chest sounds, or you can image that chest to see if there's fluid in the lungs, right? But chest tightness is not something that you as the observer can deduce, and so it's a symptom. Itching, headache, all these things, right? Sore throat, those are symptoms, all right? So that might be a good quiz question, maybe, perhaps. All right. Earliest symptoms of a disease are a result of the body's defenses, right? So fever, feeling like you got hit by a truck, swelling, that's not the microbe doing that. It's your body doing that in response to the microbe. The fever is meant to burn out the microbe. Signs are going to be things like that cause inflammation. Edema is swelling of your lymphatic systems. You get fluid, right? Granulomas and abscesses are the body's way of kind of sealing off the microbe. All right, so if I can't get rid of it, I'm just going to wall it off and prevent it from getting out and going to other places in the body. Swollen lymph nodes, all of that. These are things that the body does to fight off these microbes before it calls in the T cells and the B cells and the macrophages and things like that. Leukocytosis, that's a sign that you can measure by just taking a blood sample and looking if there's an increase in white blood cells. Or leukopenia, is there a decrease in white blood cells? Those are things you can measure. Septicemia, are there microbes in the blood in high concentrations? Bacteremia, viremia, okay? Bacteremia is bacteria present in the blood, not necessarily multiplying, okay? So viremia is viruses in the blood, not necessarily multiplying. So we have septicemia, bacteremia, and viremia. Septicemia is when you're in bad state, right? Large numbers present. Bacteremia is bacteria specifically, low numbers. Viremia, virus specific, low numbers. Now, bacteremia and viremia can then turn into septicemia if the body can't fight it off in time. Asymptomatic, subclinical, right? There are no signs. Maybe there are symptoms, maybe there aren't, but there are particularly no signs. All right, especially if you have, there are people that have, hypochondria. There are people that mistake symptoms of some disease for symptoms of another disease, right? So asymptomatic really should apply to not showing signs, even though we call it asymptomatic. We don't call it asinal, right? But typically, if you're asymptomatic, you're not showing signs. Symptoms are subjective, so we take those into consideration, but we're really looking for signs. Latency. After initial symptoms and signs start to wane, the microbe then becomes quiescent, okay? It stops reproducing. And so the body cools off. It says, okay, I guess I'm done fighting that invader. But what the body doesn't realize is that the invader is still there. It's just taking a break and it's hiding out incognito, and it will come back later on. Chronic carrier is a person with a latent infection who then sheds that infectious agent. You can shed an infection and not have symptoms of that infection. You can be, obviously, if you have symptoms and signs, you're going to be much more likely to be contagious, but you can still pass it on when you aren't currently experiencing signs. Sequelae, long-term or permanent damage to tissues and organs, okay? So we can have damage to our tissues and they heal. And then that's a thing of the past, right? But sequelae is going to be long-term, pretty much irreversible damage. And that's what's freaky about SARS-CoV-2, right? With COVID-19, it's a new virus. We don't know if there's sequelae or not. And signs show that there might be. There are these long haulers that are suffering months after being infected, months after being negative for the virus. They're still experiencing symptoms. What's the damage it has to your heart? You know, we don't know these things. What is the long-term lung damage, right? And so for me, particularly with kids, my worry is that it's not like it's going to, it may not kill them, right? Because kids seem to have less of a hard time with COVID-19, but maybe there's sequelae. Maybe it damages their lungs irreversibly for the rest of their life. You know, and they don't die, but they deal with a lifelong damage. So sequelae. Reservoir, that's going to be the primary habitat of the pathogen. Okay. Often it's going to be, if it's a zoonotic infection, it's going to be an animal that is the reservoir. Bats are great reservoirs of viruses, okay? Soil, water, plants. The pathogen usually doesn't have a human as a reservoir, okay? It has something else, and then it leaps into the human, wreaks havoc for a bit, and then, you know, dies down. Source is the individual or object from which an infection is actually acquired. All right. So let's say COVID-19, from what we can understand, the reservoir was a bat that then jumped into a pangolin and then jumped into a human. So what's the reservoir? It's a bat. What's the source? A pangolin. What's another source? Another human. Okay. And so now, pretty much the source is humans. But the reservoir was initially the bat, and they think that this virus has been around for a long time in that reservoir and just didn't make that leap yet. In terms of carriers, you can have asymptomatic carriers, like I said, no signs or symptoms. You could have passive carrier, and that could be a contaminated healthcare professional that is transferring that disease or that microbial agent that causes disease from one patient to the next, right? Let's say it's a male physician who's wearing a tie, and then that tie touches the bed that the person coughed on, and then they go, isn't infected. They're not the kid from better, feel good. So if they go back to work, literally the spread, shut it. It's vectors, whether it's mosquitoes, actively participating in the current, not sterile, and tell me about how infect autoclave, the human, zoonosis, in fact, get rid of dengue or Zika virus, right? If you don't get rid of them, you don't get rid of the disease. Here is a great example. This is my son, and this is my dog, and he's eating his macaroni and cheese, and then he's feeding some to the dog with his spoon, and then he puts the spoon back up to his mouth, and luckily, he seems to be fine now. But this goes back to the microbiome question that Emily had. I'm sure after that endeavor, the microbiome of my dog Clyde and my son Theo was more similarly aligned after this event. But that's how it happens, right? In contact with animals. And luckily, that makes us different. Animal interested in this, I hold you accountable for this information. But all right, we're almost hospital. Person in us, cut places. Okay, yeah. So the most breathing, like 2 to 4 million cases of disease that were acquired in a hospital, resulting in 90,000 deaths. And this is a table. I'm not going to make you guys, again, don't focus on this highly detailed stuff, but this goes into the different areas of your tissues that have potential for nosocomial infections and the prevalence. So your blood, luckily you're less likely to get, blood infections, just because hospitals are very, very cautious of bloodborne pathogens, and they try to control that as much as possible, but much harder to control something that is entered into the respiratory system. So breaks it down into the different types of microbes that affect different areas. And again, just for your information. Prevalence, we hear about these things. And I like to go over these concepts because these are things that are used a lot in common vernacular, but aren't used correctly. And you'll, going into nursing and stuff, you'll use these, right? So prevalence is the total number of existing cases, considering the entire population, okay? Whereas the incidence is the number of new cases in a certain time period, okay? So that's the difference. Prevalence is, at any given point in the US, how many cases do we usually see? Okay. Whereas incidence is, how many new cases are we seeing? Okay. So you could do the prevalence of COVID-19. That would be different from the incidence, right? Because the incidence is what we're seeing going up and down, right? We're seeing new cases surge up to 1,000 new cases and then drop down to maybe 800 new cases and then surge again. That's the incidence, not the prevalence. Mortality rate is the total number of deaths, whereas morbidity is the number of people with the disease. So not the number of people that die from the disease. That's the mortality rate. Morbidity rate is how many people have it, okay? And those numbers are going to be different depending on how deadly the disease is. Epidemic, pandemic, we all know these well now, right? Epidemic is more isolated, right? The prevalence is increasing, but it's restricted to a region, whereas a pandemic is worldwide, okay? Any questions on this, guys? Or comments?
Dr. Franz.
Yes.
I'm caught up on a couple slides back with vector. There's a little indentation there where humans cannot be vectors. Can you clarify that for me?
Let's see here.
So you were talking about your dog,
Yes.
Clyde, son Theo, and exchanging whatever
Yes.
saliva food, and no cross-contamination there. But when it does happen, let's say, can we be a vector to the dog and that we get the dog infected somehow?
So I'm trying to see a live animal other than human.
Other than human, I guess, is what I'm asking you about.
Yeah, so no, technically humans aren't vectors to themselves. A vector is kind of like considered a line, like a connection between two different things. So so a vector is going to be a different species infecting a different species. Now, of course, humans can transmit that disease to other humans, right? But we're not considered necessarily vectors. We are carriers. So kind of a semantic thing that's a little bit confusing. I get that, but by the very nature of vector, it's we are blaming something else besides us for acquiring that infectious agent.
Is Carl?
Yeah.
Is that like humans could technically be the concept of vectors? We just don't use that words for our own species because we use that words in relevance like to ourselves and what gives us diseases.
We can be vectors to other organisms other than ourselves, but we aren't technically defined as vectors to each other. Okay, we are carriers. So if it's a vector, it's two different points, and those points are different species. Thank you for clarifying, Ryan. That's a good point. Okay, anything else? Any other questions?
I got one on just a personal note with domestic animals, and you know, slowly this class is creating a germaphobe out of me. So what is the house pet of choice from a microbiologist's point of view?
Oh, I mean, I have a dog, so
Sure.
that I tend to think that I wouldn't worry about animals. You know, I think that generally, if you give your dogs heartworm and stuff like that, they'll be okay. They don't really tend to bring in they are not good vectors for humans. Now, if you are bringing in, if you're going to be like the wildlife center and bring in some injured robin into your home, might not be a great idea, right? But generally speaking, we don't see outbreaks by these normal typical pets. We're seeing zoonotic outbreaks from insect arthropod vectors primarily. And then things like emerging viruses in these wet markets where animals are put into proximity to each other that were never placed in proximity before. And so you see particularly viruses jump around that way. But luckily, most of our pets don't bring in stuff that will harm us. Now, that said, you shouldn't let a dog lick your wound. The whole concept that they have like antibacterial saliva is not something you want to adopt. In fact, their mouth is riddled with even more bacteria than ours. And we just learned how much bacteria we have. And if you let them go licking your wound, that can, again, a wound is a break in your protective barrier that can lead to all kinds of terrible systemic infections. So that's my recommendation from a microbiologist standpoint. Have your pets. I think pets are great. And they help us with our mental point of view. And as we said, mental stress can affect your immune system. So if they make you happy, by all means, have them. Any other questions? Okay, let's take another 10-minute break. We'll start at 12:05 and then we'll do our journal club, okay? I know this is a lot of sitting and listening, but now it's your turn to talk. I'll try to talk minimally so that, you know, you don't have to sit here and listen to me for 3 hours straight. So we'll take a break and we'll be back at 12:05.